So we've spent our time so far this semester talking about how we get energy from glucose. Now we're going to talk about how we get energy from fat. Lipid metabolism. So we're going to talk about how when we eat fat, is everybody okay? So when we eat fat, what do? We have to first hydrolyze the triglycerides, get that glycerol off, and make our fatty acids. Then what we have to do, because beta oxidation happens in the matrix, we have to get it into the matrix. And it's not simple. In fact, I think it's silliness. It's kind of weird what the cell does to get that fatty acid into the matrix where beta oxidation happens. So we're going to talk about getting it past the two membranes, strangeness. And then we're going to talk about beta oxidation, which is super cool. It's very simple. We're going to compare, and we've talked about this before, how we get more energy, carbon per carbon, out of fats than we do out of glucose. And then we're going to talk about the oddballs, odd number fatty acids, what do we do with those, and unsaturated fatty acids. How do we handle that? Then we're going to talk about another class of molecules, ketone bodies, where they result from. And it's really from having too much Acyl-CoA. And then we're going to talk about oxidative stress. So let's talk about fats. Fats are the primary storage form of energy. All right? Why would they be the primary storage form of energy? What do you think? I already said it. You know, but why do you think you can get more energy from a fat than you can from a carbohydrate? Think about the structures. Look at the carbons on your fatty acids compared to the carbons on your glucose. There's more. It's not. But carbon for carbon. If you compare the one carbon from, or the same number of carbons from a fat, it doesn't matter. So what is the process of getting energy? What are we doing? We're burning, right? We're going to carbon dioxide, right? And we said that metabolism, instead of burning, we're slowly burning. You're slowly oxidizing each carbon to carbon dioxide. So, so the process is still the same. So what we have is like a ladder. The top of the ladder is carbon dioxide. And each step up, instead of lighting it on fire to burn the glucose or fatty acid to get the energy, the product is the same, carbon dioxide and water. But what we're doing is we're doing a slow burn. And each time you go up a rung of the ladder, you're capturing energy. When you start out with a fatty acid, most of the carbons, except for one, are all carbon-hydrogen bonds. When you start out with a carbon on a glucose molecule, it's carbon-oxygen already. Carbon dioxide is carbon with two oxygens. So here's our ladder. All right. The top of our ladder is the ultimately oxidized. So this is a C double bond O. Let me write that here. C double bond O. So here we are at the top. C double bond O. When we have just carbon, hydrogen, another carbon, we're basically at the bottom. When we have our carbohydrate, we're already partway there. So we're going from carbon, no oxygen bonds, to carbon, two oxygen bonds in metabolism. That is the process. But when you start out, so here's a fat, and this is a carbohydrate. When you start out with a carbohydrate, you're halfway there already. Maybe not that much. I put it at half the middle. I would say it's maybe one rung up, But you're already there because we've got to get to the double bond there. So we're starting up higher in the ladder. We have fewer steps to get to that C double bond O, to the carbon dioxide. Here with the fat, you have more steps to go through to get to the carbon dioxide. So each step, each time we go up the process of getting to carbon dioxide, you're going to get energy out each time. So the more steps you have to get to carbon dioxide, the more energy you're going to get out. Because we're already up on that ladder with carbon dioxide, with carbohydrates, we are going to get less energy out.
Can you say that last part one more time?
So every time you go up a step of the ladder to get to carbon dioxide, you're releasing energy. We're starting at the bottom. We have a reduced carbon. This is reduced carbon. This is oxidation, to go oxidation or burning. All right? So to get the energy out is an oxidative process. And what we're doing, if we just burn it, we're going from the carbon reduced form to the ultimately oxidized carbon dioxide. So here, what we've done is we've already had a little bit of oxidation go on with our carbohydrate. So there's fewer steps to get through in order to get to that carbon dioxide. I don't know how you're going to write that down.
No, it's okay. I needed to actually listen.
Yeah.
So you're saying you get more energy out of the fats because
you have more steps to go up.
Steps, you're releasing more energy.
Yeah. Because in order to get from carbon-hydrogen bonds to carbon to oxygen double bonds, you're starting at an earlier starting point. Here, you've already got a carbon-oxygen bond. So you've got a head start. But you're not going to gain as much or give up as much energy because of that head start. So think every time you climb a rung of that ladder, you're releasing energy to get to your carbon dioxide. So I said the carbons in the chain of our fats are highly reduced. Remember, carbon-hydrogen bonds are a reduced form of carbon. Carbon-oxygen bonds are the oxidized form of carbon. Carbon dioxide is the ultimately oxidized carbon. That's why it's the production of burning. Burning is the same thing as oxidation. When you burn, you react with oxygen. So this is an unfair comparison. But we'll do another comparison later. So glucose only gives you negative 15.9 kilojoules gives off, so that's why it's negative. And palmitotic acid, which has 16 carbons, gives you off 38.9. So, but we're going to compare it later, all right? We're going to do a better comparison later because it's not the same number of carbons here. But we'll see later that we get more energy, not much more, but more energy out of our fats. Okay, so what do we say the first thing? You eat your steak and you get a triglyceride. That sounds good. Eat your steak, get your triglyceride, but what we're going to do is we need a fatty acid out of that. So what do we have to do? We have to cleave the ester bonds from the glycerol. So what we're going to do, remember I said esters are little old weak ladies. They're very susceptible to hydrolysis. So we use lipases to get, and water, to get our glycerol and our fatty acid. We can either reuse it to make membranes or whatever we need to do, or we can go into beta oxidation. Now, if you have a phospholipid, here, or a phosphatidylcholine, whatever you have, if it's got a phosphate, we're going to use a different enzyme. So you need different enzymes, actually, to break apart different parts of our triglyceride. Here we have a phospholipid, and we can see like the N1 would need one enzyme. Here the middle guy would need a different enzyme. So there's a lot more going on than what I'm telling you. That's all you need to know from this slide. There's a lot more going on. All right, so we have to get our fatty acid. So we've eaten our steak, gotten our triglyceride. our enzymes in our intestines have broken it up, and now we have to get it into the matrix where beta oxidation is happening. How do we do it? We cannot get a fatty acid through the membrane. So what do we have to do? Well, remember, just like with glycolysis, you have to put in energy to get energy out. This is the same thing. What we have to do is put in energy. Actually, we're going to count this as 2 ATPs because we're giving off 2 phosphates. We're going from an ATP to an AMP. So we're going to count this as 2. I know, it's kind of weird. We're going from an ATP to an AMP. And so mono, adenosine monophosphate. Adenosine tri to mono, 3 to 1. And this PPI stands for 2 phosphates, 2 inorganic phosphates together. All right, so you put in that energy, you get your fatty acid with your AMP, and then you react it with a coenzyme A. Remember, the sulfur part of the coenzyme A is the one that does the work. The AMP, what it does to the fatty acid is it polarizes this carbon so that it will be more partially positive, so the sulfur can nucleophilically attack it. So then you get Acyl-CoA. An acyl group is the carbonyl with a carbon chain on it. An acetyl group is it's just one carbon. So an acyl group this is an acetyl group. So C double bond O/R, meaning like a long chain. I'm sorry, you can't see it. Long chain. That's an acyl group. C double bond O connected just to one carbon. That's an acetyl group. So this is an Acyl-CoA. So we need to put in energy to get the coenzyme A on. So here, this is the overall reaction. This is a thioester bond, and this takes place in the cytosol. Okay, so these fatty acids, if they are stored as triglycerides in your fat tissue, it will be released in response to a hormone binding to the surface of the cell. Okay, so here, that gets you through the outer mitochondrial membrane. The coenzyme A will get us to the outer mitochondrial membrane. Beta oxidation happens in the matrix. So now we're in the inner membrane space. The acetyl-CoA or the acyl-CoA cannot get through to the matrix. It cannot get through the inner membrane. The outer and inner membrane of the mitochondria are different. So the outer membrane is permeable to the acyl-CoA. The inner membrane is not. Why, I don't know, but it's not. What we have to do is we give it to, that acyl group is given to carnitine. Carnitine then can carry it through the inner membrane. So we take in the inner membrane space, we have carnitine. Here I'm just showing you 2 different structures. This is carnitine, and this is also carnitine. Okay? I'm just showing you the structure in a different way because it's sort of hard. Carnitine is both positively and negatively charged, so I think that's what helps it get through. So you give it up from the coenzyme A, you give it to carnitine, now it can go through to the matrix, crosses the inner. All right, so this is where we're at. Let's take a look at this picture. Here's the outer membrane, right here, and here's the inner membrane. So what we have is our fatty acid, and it gets put together with our coenzyme A in the cytosol, and now we have our acyl-CoA. It can get into the inner membrane space. This cannot get through here. this inner membrane. So what we have to do is it's going to be reacted with carnitine. The coenzyme A gets released and go back to be reused. This acyl-carnitine, so acyl group from the fatty acid, carnitine over here, it's going to help it get through the inner membrane. But what are we going to do next? This is why I think it's kind of weird. We're going to give it back to coenzyme A. It was just with coenzyme A. Why do we do that? Why do you think we're going to do that? We have to give it to something that can't get through the inner membrane. What if the membranes were permeable to coenzyme A? It would just go back into the cytosol, right? That's why you do it. So it's kind of weird, I think, until you think about it long and hard. Why do we give it to coenzyme A, then give it to carnitine, and then give it back to coenzyme A? Because if both membranes were permeable to coenzyme A, then we wouldn't have beta oxidation happening. It would just go back, right? It would just go back into the cytosol. So we need that carnitine in order to get it through the inner membrane. And now we're ready for beta oxidation. And I cannot.
What causes the Acyl-CoA to...
All right, I'll go back to it.